Hi, thanks for having me at ISMB 2020. My name is Shunfu Mao from Electrical Engineering Department at University of Washington. Today I'm going to talk about CellMeSH, probabilistic cell type identification using indexed literature. CellMeSH targets the application of single-cell RNA sequencing. Consider there are two samples, each contains several cells. Using single-cell RNA sequencing technology, we can get the gene expression at a single-cell resolution for each sample. This technology has a wide range of application. For example, consider sample 1 is before drug treatment and sample 2 is after drug treatment. Based on the gene cell expression matrix, we can estimate the cell type composition for each sample, and understanding the cell type composition difference between the two samples can help us understand whether the drug treatment is working properly or not. But to enable such kind of application, it requires accurate cell type identification. A typical single-cell RNA sequencing workflow is as follows. Starting from a sample of cells, we do experiment and data processing to get a gene cell expression matrix, and we do clustering to cluster the cells with similar gene expression patterns, and we do differential expression analysis to get for each cluster differentially expressed genes, and we want to annotate for each cluster a cell type. Existing methods mainly contain the database part and query method, and they take input of differentially expressed genes for each cluster and return a list of candidate cell types for each cluster, and their databases with gene cell type information are mainly constructed by manual literature survey or from existing experiments, but restricted to manual literature survey or from existing studies. So these databases are not comprehensive, and more critically, the gene cell type information is also laborious to update. We developed CellMeSH to tackle the cell type annotation problem. CellMeSH is essentially a web server and Python API. It takes input of cluster differential expressed genes and output a list of predicted cell types for each cluster. And similar to existing methods, it contains a database part and a query method part. The database contains gene cell type information collected from prior literature, especially the indexed literature, and we designed a novel probabilistic query method in order to better query the database. We want to build a database that is rich in cell type and marker gene information. Ideally, we want to collect this information from raw PubMed articles. But we noticed there is Medline database, which contains references for almost all PubMed articles, and each reference indexes mesh terms for the article. Mesh is a controlled vocabulary in tree structure that contains important biomedical concepts, including 570 cell types. We also noticed there is Gene2PubMed database that indexes important genes to related PubMed articles. So we want to associate the genes indexed in Gene2PubMed and the cells indexed in Medline to create a database that is rich in gene cell type information. The CellMeSH database is a species-specific we create for both human and mouse. Essentially, the database is gene cell matrix, where the count means the number of PubMed articles, where the gene is indexed in Gene2PubMed, and the cell type indexed in Medline. The CellMeSH database has large coverage of gene cell type information. It is also easy to expand as new literature come up, but it has publication bias. Some common gene and cell type, they tend to have larger counts. And there is also spurious gene-cell association because the simple fact that a gene and cell type co-occur does not mean the gene serves as a marker for the cell type. Therefore, we need novel database query method. Our probabilistic query method first pre-processes the CellMeSH database. In particular, we do TF-IDF transform. Each gene cell weight is multiplied by log NC over kg. NC is the number of total cell types, and kg is the number of non-zero counts for a particular gene G. For common genes, the kg tends to be large, and the related weights will be penalized. Therefore, TF-IDF essentially addresses the publication bias of common genes. We also do column normalization, so the weight will be adjusted by the column sum to address the publication bias of common cells. Then our probabilistic query method will take input of the weight-adjusted CellMeSH database and also a query of a set of genes and return a list of candidate cells sorted by probability of query given the candidate cell type C. So this score is calculated using maximum likelihood estimation and addresses the spurious gene-cell association. For details, please refer to our poster. To check the cell type annotation performance, we first use human PBMC dataset, which is essentially a gene cell expression matrix. From the original publication, we already know the cell clustering, and for each cluster, we have the annotated cell type. We use 1 versus rest to get differential expressed genes for each cluster, and for each cluster, we prepare the query. We use the already known annotated cell type as a reference cell type for the query, and we keep top 50 differential expressed genes as the query set of genes. We use these genes for each query to query CellMeSH and get results. And we compare the results with reference cell type using heat map. This figure shows the PBMC annotation results. Along the y-axis are the reference cell types for the queries, and we show a subset of mesh cell types as candidates along the x-axis. For each query, we use red, yellow, and blue color represent the first, second, and third retrieval results. And if the retrieval result is correct, we highlight in border box. For example, for B cell, we get first and second retrieval results as B lymphocytes and T lymphocytes. Because B lymphocyte is the correct one, we highlight in border box. Because T lymphocytes is not the correct one, its yellow color turns lighter. And as we can see along the diagonal border boxes, almost all are filled with color, indicating that almost all the queries get a correct answer within the top 3 retrieval results. There are also vertical bordered boxes indicating these queries, these reference cell types, have the same expected mesh cell type at a coarse level. This is due to a resolution limit for the mesh cell types because we did not find fine-grained terms that can better describe these reference cell types. For the query with reference regulatory T cell, the expected mesh cell type should be T lymphocytes regulatory, but it's not colored. As we can see among the top retrieval results, this expected mesh cell type actually ranks 6th, but the top 3 retrieval results are closely related to the expected one. For example, the first result, T lymphocytes, is actually the direct supercategory of the second result, CD4 positive T lymphocytes, which is in turn the direct supercategory of the expected result, T lymphocytes regulatory. Therefore, the top retrieval results are actually still reasonable and promising. To further understand the CellMeSH annotation performance, we compare with existing systems using top K accuracy. We use 3 mouse datasets, which are essentially gene cell expression matrix. And from the publications, we also know the cell clusterings and each cluster the annotated cell type. And for each cluster, we use 1 versus rest to calculate the differential expressed genes. And for each cluster, we prepare a query. We use non-annotated cell type as a reference, and we keep top 50 differential expressed genes as query genes. We query CellMeSH to retrieve the results, and we compare with reference to calculate top K accuracy. Top K accuracy means the percentage of queries that get correct answer within the top K retrieved results. Similarly, we query with other systems to get the results, and compare with reference to calculate top K accuracy for comparison. These two figures show top 1 accuracy and top 3 accuracy respectively. We compare CellMeSH with existing systems including Enrich, CellMarker, and scQuery. We also include random retrieval as a lower bound. In each figure, we have 3 groups with respect to 3 mouse datasets, and in each group, different systems are compared. For example, in Taiwan-Maurice droplet dataset, CellMeSH achieves about 60% top 1 accuracy. This means among 60% of the queries, their top 1 retrieved results are actually correct. And CellMeSH is consistently better than other systems across different datasets. And this trend is similar for top 3 accuracy. We have open sourced CellMeSH as Python API at GitHub, including the database and the query method, so it can be integrated into our existing single-cell RNA-seq analysis workflow. Actually, we have integrated CellMeSH in our ongoing work, the OncoAPP, which is a web server that supports online graphical user interface to do single-cell RNA-seq analysis. And it supports automatic cell type annotation and also iterative analysis. For example, starting from a gene cell expression matrix, we can do pre-processing and clustering, and we can do dimension reduction to visualize the clustered cells, and we can do differential expression analysis, and we can use CellMeSH to take input of cluster differential expressed genes and predict cell type for each cluster, and the user can visualize the clustering and annotation results and split and merge the clusters and redo this whole analysis iteratively in order to refine the analysis results. CellMeSH has a standalone website under OncoAPP web server, so lab scientists, they can do their experiments and input the cluster differential expressed genes or simply a set of query genes, and they can select the CellMeSH database, or there are other databases such as CellMarker for additional options, and they can select the probabilistic query method and species, and CellMeSH will return a list of candidate cell types, their IDs, probabilities, and overlapping genes between the candidate cell type and the query genes. And for each overlapping genes, the PubMed articles where the gene and the candidate cell type co-occur will also be displayed for further reference. As a summary, we have developed CellMeSH to tackle the problem of single-cell RNA-seq annotation. CellMeSH takes input of a set of genes, for example, cluster differential expressed genes, and output a list of mesh cell types. It contains a database part and a query method part. The database is built from indexed literature, currently only for mouse and human species. The database contains large-scale gene cell information, and it's also easy to expand. But the database is inherently noisy, so we designed a novel probabilistic query method to better query the database. CellMeSH has shown superior annotation performance, and we have provided its Python API and web server so it can be used by bioinformaticians and life scientists. But it still has limitations. Some query results are still coarse-grained, probably due to a resolution limit from mesh cell types, and the database is not sufficient for other species, probably because there are not enough publications and not enough signals. In the future, we plan to use natural language processing technique to extract cell type and marker gene information from raw text directly, and we want to incorporate extra mesh information to provide more comprehensive view for the given set of query genes.